All right, just a quick review. First and second law of thermodynamics. Here we have a cheetah. It's devouring a gazelle. It's transferring the chemical energy of the gazelle into the cheetah. Then it wants to go run after another gazelle. As it's doing that, it's taking the chemical energy in its body and converting it into mechanical energy, giving off heat, which is very similar to entropy. And then the carbon dioxide and water, they are also very disorganized compared to the molecules that they were before, whether that's glucose, which is a large, 6 carbon sugar that gets broken down into a bunch of little carbon dioxides and water. Okay, so we're creating disorder and disorder. Living cells, you can't get away from making heat. All right, if you want to, keep the main functions of a cell, just you're going to continue to make heat. You're going to make disorder. Anyways, different processes that occur without any sort of initial net energy input, we call these spontaneous. Then we have non-spontaneous. Anyway, cells, what they do is they create order. They are ordered, okay? And what we do as living things is we end up, making like disorder, but then we kind of like shuttle it out of the cell because we want to maintain our order, okay? And what that does is it kind of creates an increase of disorder outside of ourselves. That's the expense, but we stay ordered. All right. So one of the things that is important for like organisms is to be able to, kind of do a number of like transformations of chemical energy into mechanical or other types of energy, or we want to be able to take different chemicals in our body and change them into one type or another. And that's even though there might be spontaneous processes that don't need extra energy or may need some, this doesn't necessarily happen instantly. Well, one of the things that can speed that up are enzymes, which are just catalysts in the kind of like biological world. And by the way, a catalyst is something that speeds up reaction. It's not consumed by that reaction. So enzymes are just proteins that are catalysts. So here's an example. We have sucrose, which is table sugar. We add an enzyme, sucrase. Notice the ASE suffix. That tells us that's an enzyme. Anyway, sucrase will break sucrose down into glucose and fructose. All right. It also has to expend a water to do that. Well, pretty much all reactions, whether they're spontaneous or not spontaneous, have this activation energy kind of barrier, and that is this initial energy that has to kind of be given to kind of kickstart the reaction. Think about your lawnmower. If you don't have an electric starter, you have a pull starter. You got to pull the cord on a lawnmower, and you get that initial energy kind of input, and then you get all that back. Think about a match. Match has lots of potential energy in it, but it just doesn't light on fire. You need to strike it on something. Striking it on something adds that activation energy to kind of kickstart that reaction. All right, so here's a little example. We have a couple of reactions, A, B, and C, D. They're just hanging out. Well, in order to get them to kind of do this reaction where A and B switch with C and D, it needs to kind of destabilize the bonds between A and B and C and D. Okay, that's where we add that activation energy, we destabilize the bonds, and then the reaction can kind of continue as normal, where now A is no longer bound to B, it's bound with C. Free energy in the system is just the energy that's available to do work, that's actually available. Anyway. So our enzymes, what they do is they lower the activation energy. So we have here kind of a representation where, we have reactants and products, and our free energy, in this case, it looks like it almost halves the enzyme, what it does is almost halves the amount of free energy that we need. So it takes less time to get to the peak and then less time to get to our kind of final product. The product's going to be the same, it just takes a lot less time. Keep in mind, the x-axis is not time, it's just the progress of the reaction. But, presumably if it was time, the red line would hit bottom before the black line because it takes less time to get to the peak of our activation energy, which means it's going to take less time to get to the end of the reaction. So what do enzymes do? They lower the activation energy needed to do a reaction, and then that speeds up the reaction. So we're going to look at the anatomy of an enzyme. There's whatever the enzyme is working on is called the substrate. So that previous example was sucrose. Sucrose was a substrate. All right. Then the enzyme is going to bind with the substrate. All right. There's a site on the enzyme called the active site, and that's where the substrate actually binds. So here we've got an enzyme. It's got this little portion here called the active site. That substrate is going to fit in there, and the active site might actually kind of change shape a little bit to kind of help that fit. It's called the induced fit model. Anyways, the substrate is going to hit that active site, and then whatever reaction is going to occur will take place, and then it moves on. The whole idea, though, is that the substrate doesn't know to go to the active site. It's just kind of moving at random, and then if it coincidentally hits the active site, bingo, we got a reaction. So the activation site can actually lower the active site can actually lower activation energy by either just kind of orienting the substrates correctly. Maybe they wouldn't necessarily hit each other in the right way if you have two substrates, or it actually might strain the bonds within the substrate, kind of destabilizing them. Might just make a more favorable environment for the substrate to react. Or it actually might just straight up covalently bond to the substrate to make something happen. So we're interested in things that can affect the enzyme's activity or rate of activity, one of which is substrate concentration. If you have a lot of substrate, you would expect that there's a higher chance that substrate is going to hit the active site and then the reaction will occur. We might have environmental factors which are important, like temperature and pH. And then we might have other chemicals that specifically influence the enzyme, which we'll talk about in a minute. So each enzyme is going to have an optimal temperature and an optimal pH that it's going to function at. So for instance, our body temperature is approximately 37 degrees centigrade. So we expect our enzymes to actually function really well at 37 degrees centigrade. The pH in our stomach is 2. We expect the enzymes in the stomach to function really well at pH 2 versus our small intestines has a pH which is a little bit basic, around 8, and the enzymes in the small intestines function really well at pH of 8. Couple of reasons for temperature. So temperature will actually speed up the substrate and we'll get it to increase the probability that it will actually hit the active site. But at a certain point, when you heat it up too much, you'll denature the protein. And same with pH. These enzymes, they function well at certain pH. If you change the pH too much, you're going to denature that protein and you're going to kind of distort the active site. It's not going to work so well. And just up on as far as temperature goes, like for us, our enzymes really work well at 37 degrees. But there's certain heat tolerant bacteria that live in geysers or whatever, their optimal temperature for their enzymes might be around 80 centigrade, which is really, really hot. There are these things called cofactors. They'll kind of pop in here and there, but basically, we have things like coenzymes, which are vitamins that just kind of help reactions along. Cofactors are usually metals that kind of help these reactions along. Basically a lot of things in your daily vitamin are cofactors. You don't need a whole lot, but you still need them. We have things called inhibitors, and they slow down the reaction rate. We have things called competitive inhibitors and allosteric or non-competitive inhibitors. Competitive inhibitors are actually shaped similarly as a substrate, and what they do is they bind the active site, and then they kind of block the substrate from getting in there. Allosteric inhibitors, what they do is there's a separate site other than the active site called the allosteric site. So an allosteric inhibitor will bind the allosteric site, and it'll actually change the three-dimensional shape of the enzyme, thus kind of shutting off the active site. So here's kind of a drawing. We have normal situation. Substrate hits our active site right here, makes the reaction happen. Well, in this case, our non-competitive inhibitor kind of blocks our substrate, so then our substrate bounces off. Whoops, not going to happen. And then on this far right example, we have an allosteric inhibitor is hit the allosteric site here, and you can see that it's slightly changed the shape of the protein, but doing that, we're not allowed. We don't have the reaction doesn't occur, so that slows down the overall rate of reaction. So allosteric regulation is not like a horrible thing, because that gives the opportunity for the your body, your cells, to turn things off and also turn things on. Or we get kind of like a promotion. So anyway, we have something called an activator, where it kind of hits in there and kind of turns everything on. But our inhibitor can also kind of land in there and turn it off. So it all depends. But this is one way in which your body or the cell can regulate enzyme activity allosterically, either turning on or off the active site. Yeah, that's it.